Hello, everyone. This is Braswell Dean, along with my co-host of last night, Dr. Haley Brownstein, giving you a re-recording of the Highland Park Metabolic Support Group meeting that we had last night. I guess it's not a re-recording; it's a recording. But our meeting was last night, and that was April the 11th of 2024, and I forgot some of my handouts last night, as I'm prone to do sometimes. So I thought I would give you guys just another rendition of last night, and maybe this would be helpful to you guys as well as maybe people who join the group in the future. Like we said last night, we were welcoming the new members, and we hope you find a good place to connect. And as always, we wanted to say thank you to Highland Park Church for allowing us to meet in this wonderful facility, and all the goodies and snacks and things that you've provided is much appreciated, and as well as your prayers. Why do we do a group? Well, as we said, research will show up and down that there is tremendous amount of strength in numbers, and the encouragement that you get, the camaraderie, as also the learning that you get is very, very powerful. And for most, it can be stronger than whatever medicine people are prescribing, or what's in a medical textbook, or treatment protocols, whatever. It's the relationships that really carry the day. So we wanted to maximize that, and we acknowledge that this group, the strength of this group, more than anything, are each individual person here. Each individual person is a hero to us, and we just see it's just a great way of giving you something that you just can't get. If someone has a cancer, and it's like it can be isolating in many respects because nobody understands. Even people in your own family might not know what you're going through, and you might have a variety of emotions you're going through. But when you connect in a group like this, you're going to find people going through the same thing that you can connect with on a deeper level, and it can be very, very helpful in so many ways. We're not claiming that anything that we say is a cure for cancer. We're not saying that. We're not saying we know everything. We don't have crystal balls or anything like that. But what we can claim, what we are claiming, is that you getting better is everything to us. Everything. So in many regards, we are truly learning together. We're encouraging one another, and we are trying to connect with one another in varieties of ways. So anyone can come. We're inviting anyone to join this group. You don't have to be a member of the church. You don't have to be a patient of mine or Dr. Haley Brownstein's or anything like that. We're welcoming anybody. You can have friends or family come. What we do ask, though, is you work with your primary doctor, either me or Dr. Haley or your own doctor, and certainly your own oncologist. We're 100% respectful of your primary doctor and your oncology doctor and what they're telling you to do. We are not giving individual medical advice. This is a group discussion, so you can't take what we give and apply it directly to you. And certainly if you're looking at this video and you say, oh, okay, I'm just going to do what they say, that's not what we're saying. You want to learn from this and then apply it working with your doctor. So that's how we feel about that. And as far as sharing in the group, obviously any group format, the information that you share is designed for you to share as you feel comfortable. You can do more talking or more listening. It doesn't It's up to you. But in our group, when we meet as a group, we certainly hold everything confidential to the group, and we feel like people are sharing to enhance one another, encourage one another, and also get things out of it yourself. It's okay to have different opinions from other group members, or it's okay to have different treatment strategies. People are going to read different things and want to do different things different ways. That's fine. And we acknowledge that not everyone's plan is going to look the same. So there's that. There's some introduction comments. So again, I titled this talk Brief Concepts in Cancer because we certainly have more information that we can go into, but I wanted to keep this brief, like an overview, especially when new people join the group. And certainly our existing people can use things like this maybe to have a reference. Dr. Haley Brownstein and I thought a few years ago that our cancer patients just seemed to get lost in the medical world of oncology treatments, and we just hated to see it, and we wanted to help in some way, give them something more, something that we could help with. So that's how this started. It was really a humble and sincere desire to help our patients. So let's start off. This is from the FLCCC Alliance, and it basically is a slide that outlines sort of the modern day traditional cancer treatment options, everything from hormonal therapy, surgeries, bone marrow transplantation, chemotherapy, targeted therapy, radiation, immunotherapy. All of these therapies have something in common, and that thing in common is that the main theory is that cancer is thought of as starting from a genetic mutation. Interestingly, that this mutation theory started sometime after Watson and Crick devised and came up with the DNA model and understood what DNA was, and began to unlock a whole new frontier in molecular biology and molecular biochemistry and genetics, and it just created this new horizon that people were heading towards to try to eradicate cancer by coming up with the genetic code for the mutation and then creating a drug that could target these cancers. And so that started this journey, and we see a lot of different drugs now being developed for cancer. It started off in the tens of millions. Now it's hundreds of millions of dollars now are spent by drug companies to create these drugs just for one drug to target a genetic mutation. Interestingly, recently, Dr. Watson, I believe it was Dr. Watson of the Watson and Crick team, he came back and circled back, and now that we have computers helping decode the human genome, he's been quoted as saying something to the effect that it's too complicated. The gene code is too complicated to try to continue to develop drugs against a mutation for someone's cancer that is constantly mutating. So interesting there, but we are continuing to do everything we can, and certainly the gene mutation is the theory of the day. Let's look, though. It wasn't always this way. There was a fellow named Dr. Otto Warburg back in the 1930s and in the 1940s. He wins a Nobel Prize for basically coming up with another theory about cancer, and it's more of a metabolic theory of cancer. And it's shortly after World War II, his research was kind of abandoned, and then it certainly was abandoned after Watson and Crick's discovery of DNA. But he was saying that cancer really has a problem that is more of a mitochondrial problem, not something within the nucleus. So let's take a quick look, and this is a very, very simple overview of this this theory of a metabolic problem. And we look at a normal cell over here, and we contrast this with a cancer cell, which has sort of a blue cytoplasm. So let's take a quick look at the normal cell. So we look at a normal cell. It's got a nucleus right here. This is kind of like you could think of the nucleus is where the boss man of the cell lives, with all the blueprints and directions to tell the cell what to do. And this yellow thing is the cell, and the cytoplasm of the cell you could think of as like a parking lot in the middle of the cell. And this thing over here we'll call a mitochondria, and that's sort of like the factory. So you have a factory, and you have a parking lot, and then you have this other area, this nucleus, where the boss lives in a separate building over here. And the normal cell generates energy to live mainly in the factory over here. So when the factory has oxygen, it will make a ton of energy for the cell. And it is generated by what we call oxidative phosphorylation, and it generates a whole bunch of energy, and we'll call that ATP. But when the cell doesn't have a lot of oxygen, well, there's an emergency. The cell has another way of generating energy. So when you're exercising and the oxygen levels may not be so great, the cell can generate energy out here in the cytoplasm, out here in the parking lot. It's almost like the workers in the parking lot, they can't get into the factory, so now they're burning paper and tin cans out here, generating energy much less efficient, okay? So this is glycolysis out here in the cytoplasm. So preferentially, the cell wants to burn energy in the presence of oxygen in the mitochondria, in the factories. We'll contrast this with a cancer cell, and it's very, very interesting that cancer cells, this is what Otto Warburg discovered, that the cancer cells have something in common. They're not very good at all of using their mitochondria. Even in the presence of oxygen, the mitochondria of the cancer cells preferentially burn and make energy out here in the cytoplasm, out here in the parking lot. Okay? So even in the presence of oxygen, we are going to use the anaerobic, we're going to use glycolysis, which is the old-fashioned fermentation, even in the presence of oxygen. And we call this aerobic glycolysis out here in the cytoplasm. So it's almost like the workers can't get into the mitochondria. Maybe they can't. You could think of it as the workers can't breathe when they're trying to work in the factory. The factory is closed, or it has very little oxygen being used in the factory. The energy has to be created out here in the mitochondria. So you're probably saying to yourself, and we were joking last night, you're probably saying, well, who cares? Who really, really cares? Well, it is important because we want to find out we want to find out the vulnerability of the cancer cells. So if we take a deeper look in the cancer cells from the metabolic problem, from the sugar perspective or from energy perspective, and one of those energies is sugar, we know cancer cells are very, very dependent on sugar. For most cancer cells are. When glucose comes into the cell, the yellow again is the cytoplasm. Here, the parking lot. We have the factory over here in the gray. When glucose comes into the cell as sugar, the cell has to do something with it. So it converts it to pyruvate, which is another form of sugar, and then the cell has to make a decision. It either takes that pyruvate and puts it in the factory into the form of acetyl-CoA, and it goes through the factory in terms of the Krebs cycle and the electron transport chain for this oxidative phosphorylation that happens in the mitochondria and generates a whole bunch of ATP. Or the cell has a decision to make that it's going to take that pyruvate and convert it over here to lactate using the fermentation process. Now, if there's no oxygen, it's going to make lactate, but the cancer in lactic acid builds up and is irritating. It's a waste product. So it's a much less efficient way to generate energy. If the cell is healthy, it'll make lactate. If the cell has oxygen and is healthy, it will go over here and burn energy in the mitochondria. But if the cell is cancerous, if the cell has a defect, it can't use its mitochondria, so it's obligated to burn energy out here in the cytoplasm, even in the presence of oxygen. It's going to make lactic acid. Okay? And that's called the Warburg Effect. So this discovery has been around a long time. And what are the consequences of this? Well, the consequences is this lactate or lactic acid is going to build up an acidic environment around the cancer cells. You can think of this acid buildup as waste products, and that's going to stimulate inflammation. And that inflammation is going to attract new blood vessel growth into the area because the body's going to want to say, hey, you know what? We've got infection or inflammation and waste products. We're going to build new blood vessels to come in to try to repair this process. And these new blood vessels stimulate the vascularity of the tumor, and that increases the metastatic potential. So, and just another thing, I threw this in last night. I'll throw it in again tonight. The pyruvate is shunted over here to the mitochondria by an enzyme called pyruvate dehydrogenase. And that facilitates aerobic metabolism over here. And it's almost like an emergency brake. To slow this down is pyruvate dehydrogenase kinase. That's an inhibitor. But it's interesting to me that alpha-lipoic acid inhibits the inhibitor, and therefore alpha-lipoic acid facilitates the pyruvate dehydrogenase enzyme. So in other words, the bottom line is alpha-lipoic acid facilitates aerobic metabolism in cells, and that is a good thing. So alpha-lipoic acid works to try to correct the fundamental problem in the cancer. So that's interesting to me. So here is another big concept, and we're emphasizing in this talk big concepts to try to understand. So from the another big concept is stem cells. So you can think of when you have a tumor, and this is supposed to be a tumor over here, and when you give somebody chemotherapy, the chemotherapy is designed for the genetic mutation in a lot of these cancer cells. Now, most of the cancer cells will get killed from the chemotherapy, but there are a couple of things that are happening at the same time. Number one, not all of the cells in the cancer have the same genetic mutation. You can have cells at the top having a different mutation from the cells at the bottom. And so these remnants of cells with different genetic mutations will survive. So after the chemotherapy, we have now selected out a new group of cells from this tumor area that have a potentially different genetic makeup or something enzymatically that allowed them to survive. And the other thing is to understand is that the stem cells, the cells that generate new cancer cells, are not targeted by the chemotherapy, so that what do survive are cancer stem cells, which are the things that you really have to go after to kill, and any other mutations that have survived the last several chemotherapy cycles. So this lactic acid environment with what we call angiogenesis, meaning new blood vessels coming in and growing into this area, we have inflammatory processes from the excess lactic acid and cytokines being released. All this stuff are new blood flow that comes in, and it encourages stem cell transport to another area as well as these cells. So this is sort of how we see chemotherapy. One negative aspect of chemotherapy is over time, it will drive new genetic variations and allow also stem cells to survive. So the stem cells will seed a new tumor, potentially in the same area or potentially in a new area of the body that will be more and more over time resistant to the chemotherapy. Almost like antibiotics can become resistant, bacteria can become resistant to antibiotics over time because the bacteria are not just going to sit there and take it and wait for more and more antibiotics to kill them, any more than a stem cell or a cancer tumor cell is just going to sit there and grow and not change. They are going to change over time. And so that is a concept we have to think about. Another concept is, what is an antioxidant and what's a free radical? Because we always hear people talk about, oh, I'm taking antioxidants, or oh, the cancer is generating free radicals. Well, an antioxidant in terms of molecular structure is anything that can donate an electron is an antioxidant. And anything that will take an electron, or you could think of it as like a thief that swipes an electron, is a free radical. Okay, so antioxidants are good because they donate, they keep the peace. Free radicals are bad because they're thieves, they steal electrons. So we were saying last night, everybody likes a donor, somebody who donates to a good cause or donates to a political party. I suppose that's good or bad, I don't know, or donates to the church or donates to charity. Donors are good. So donors are like antioxidants. They donate things. And the free radicals are bad. They're like thieves. They take electrons, and we don't like them. They damage the DNA. They damage the cells. So when we talk about free radicals and when we talk about antioxidants, this is what we're talking about. And like vitamin C is unique, and it's a strong antioxidant because it can donate an electron and also receive an electron. So it can act sort of both ways, and so does alpha lipoic acid. So there are some antioxidants who can perform various tasks. I believe methylene blue is the same way. So changing subjects here for a minute, Jane McLelland wrote, and I hope I'm pronouncing her name right, it might be McLelland, but I'm from Georgia, so I can mispronounce anything I want to. So How to Starve Cancer, Jane wrote this book, and we had been reading lots and lots of books and reviewing articles and listening to lectures online and things. When we saw this How to Starve Cancer book, it seemed a lot different because it had so much depth to it in terms of protocols, and it put it out there. And Jane, I think, did a marvelous thing in that she took all these different chemical pathways that we were talking about, the mitochondria being the powerhouse of the cell. Well, Jane took all these different pathways that have acetyl-CoA in common. These are all mitochondrial energy sources. And she mapped out each pathway and what repurposed prescription drug or repurposed over-the-counter medicine or repurposed vitamin type thing could influence these pathways. And it's amazing that she put all this together And this is why it's so important to understand the root cause of cancer. Because if we understand that the root cause, according to Otto Warburg, might be more of a metabolic problem with energy generation, then we can devise things to try to attack it, try to starve it, and try to kill it. And when we do this, research shows that these things make the chemotherapy much more effective. And these things can minimize side effects of chemotherapy. So if you are thinking about chemo or you're taking chemo or something like that, you want to pay attention to this. And if you've been told that the chemo might not work for you anymore or that they've run out of things, this is something you want to pay attention to also, because Jane has really shown that we can have a systematic approach to a lot of these areas of energy generation that the cancer depends on. So this is another breakdown of the glucose pathway, which you think of as sugar, the glutamine pathway, which are amino acids. You can think of that as kind of a protein pathway, and then fatty acids pathways. And then Jane has listed different things on these pathways. And to be honest, there are other signaling pathways that we haven't included because there are just so many. But these are the main ones that we have listed. And I think it's really helpful to get the big concepts down before we talk about other little variations on this. So to try to keep this simple, looking at this diagram up here, cancer has fuel sources: glucose or sugar, protein, and fats. And these are the arrows. So the cell needs them. The mitochondria will use them. What we want to do is cut these off and try to starve the cancer, because the cancer is vulnerable to it. And then this is another thing over here, like IV vitamin C or other things like this, oxygen or supplements or sulfasalazine. Some other things like this can do damage when we give it to the cancer cell as well. And we'll talk about some of those as well. Now, changing subjects, this is from the Rordan Cancer Clinic. And I know this slide is quite dense here. It's a little bit too complicated, so don't worry about that. We're just trying to get the big picture here. And the Rordan Cancer Clinic does a lot of their own research and things, and they basically have been studying people using IV high-dose vitamin C and monitoring the blood and trying to come up with what is a therapeutic kill zone of a concentration of vitamin C in the blood that can be associated most significantly with killing cancer and seeing resolution of cancer. And their goal after IV vitamin C, at about 20 minutes, is around 350 to 450 milligrams per deciliter. And that corresponds to about 20 millimoles. Okay? These are just different ways of measuring concentrations. So I took a look at this, and I've been very, very interested, mainly from a standpoint of our own patients, because I'm trying to get my head around, we've got patients doing IV vitamin C and patients who are not, but for the ones who are, I'm trying to make sure that I can have some kind of oversight to see if they're getting value for what they're trying to do. So the Rordan Cancer Clinic has an IVC protocol, and these are a few highlights from this. They start off giving IV vitamin C at 15 grams to make sure, and this is after the patient has had blood tests and screening and things like that before, to make sure they're candidates for it. So there's lots more to this, but I'm trying to give you a quick highlight. If they, at 15 grams, they will measure the IV vitamin C goal at around 20 minutes. And if the patient has a plasma concentration of 350 to 450 milligrams per deciliter, then that's it. But typically they advance. It usually starts at around 25 grams. The 15 gram is more like a test dose. If they go through the test dose okay and no problems, then they'll take it up to 25 grams. And after, treatment during this month, and sometimes they'll do twice a week or 3 times a week, but they will do it on regular intervals for the first month, and they will measure post-IVC goal at 20 minutes. And if the person is already at this therapeutic goal, then that's it. They don't have to go up to 50 or 75 or 100 gram dosages, which is potentially great for patients because you're not spending money on a 50 gram or 75 gram IV vitamin C treatment, because those can be, more expensive. If the goal is not met, in other words, if at around 20 minutes the patient's plasma IV vitamin C is not at 350 to 450 milligrams per deciliter, then they'll bump it up to 50 grams, and then they will give regular treatments there. At the end of the month, I think they do another plasma draw. They see if they're meeting goal. If not, they'll bump it to 75. They'll do the same thing. If not, they can bump it up to 100 at the doctor's discretion. But though that is I think a really rational way of doing IV vitamin C. But I think I've just been really, really interested in the Rordan Clinic's therapeutic kill zone, and we'll certainly pay attention to our cancer patients. If we can get blood drawn around 15, 20 minutes, we'll at least see if we're approaching the same kill zones that Rordan Clinic is doing. I put something in about alpha lipoic acid. There are some studies that say combining IVC with ALA, which is alpha lipoic acid, could reduce the kill zone to 72 milligrams per deciliter peak, or 9 milligrams per deciliter on an average plasma concentration. This brings it down to even something we might achieve with oral high-dose liposomal vitamin C, but that is going to be a little bit beyond the scope of this talk. I'm just putting it in here for completeness sake, and just a footnote that alpha lipoic acid recycles vitamin C and also helps maintain these levels in the tissues as long as we can. So more to come, more to talk about on that particular topic. But some of the conclusions that the Rordan Clinic generated was that IV vitamin C inhibits the angiogenesis. In other words, it inhibits these new blood vessels growing into the tumor that we don't want, that these new blood vessels encourage metastasis, and we don't want that. So IV vitamin C helps with that. IV vitamin C is preferentially toxic to tumor cells, and we're going to explain that a little bit more in another slide or two, but tumor cells don't do well when exposed to this high-dose vitamin C. IV vitamin C reduces inflammation and reduces pro-inflammatory cytokines. Remember, vitamin C is a really good antioxidant, but at high dosages, it will act as a pro-oxidant. So we're going to explain that in more detail in a few minutes. But overall, it does reduce the inflammation in the tumor micro environment, and that again helps with reduction of angiogenesis. It helps with that reducing the genetic pressure for mutation, as we've discussed in earlier slides. IV vitamin C improves the quality of life in cancer patients. They've got a lot of information on this by a variety of metrics. It lessens it's not on this slide, but it lessens side effects like nausea and hair loss, and that feeling of malaise that will come with chemotherapy. It can improve response to radiation treatment and reduce side effects of the chemotherapy. And Another footnote, I have not read the articles I've wanted to read yet to present to you, but they do list um, IV vitamin C's therapeutic effect can be increased by alpha lipoic acid in combination with vitamin K3. There is a protocol for this. I just put this in for completeness, and we'll continue to circle back on this topic as time goes on. So these are the pathways, and our patients will have like a handout in front of them, and they can go through these different pathways. We kind of have like a laundry list of, are you going to take lovastatin or atorvastatin or simvastatin or metformin or berberine or DHEA or metformin or doxycycline, ivermectin, different things. But the strategy is to pick things in as many of these pathways as you can, so you can shut down as many of these pathways for the glucose pathway, as many of these pathways for the glutamine pathway. Because remember, if we do a great job of shutting down the glucose pathways, but we don't shut down the glutamine pathways, then many of the cancers will simply shift their fuel supply lines over here to, say, the glutamine pathway. And so we want to if we're shutting down one pathway over here, we want to shut down the alternative pathways over here that cancer cells use. And certainly the same thing with the fatty acid pathways as well. This slide looks like it's a little bit on the busy side, but I wanted to have a slide in here that gives kind of a bit of an overview of some of the things that we have talked about this talk, and just kind of, give you a little something to look at. Again, this here, right here, this yellow thing here is supposed to be the cell. This little double whammy thing right here is the phospholipid bilayer membrane of the cell. Okay, there's phospholipids. So the cell is sort of engulfed in a fatty envelope. This thing over here that looks pink is supposed to be the blood supply right next to it. So it's like an artery, or you can think of it as a capillary or whatever. But the blood is water, okay, water soluble. But it's got to be able to bring things into the cell through this phospholipid bilayer. And if it's fat in nature, it can kind of merge right into the fatty lipid bilayer. Like if you're delivering vitamin C by liposomal vitamin C, it can just kind of mesh right into the bilayer and come into the cell. But if it's like vitamin C in the form of ascorbic acid, more water soluble, then it's going to have to use these channels, or you could think of these as doorways into the cell when you've got something that's more water soluble. Okay, so I guess we'll start over here on the right-hand side. We have glucose in the blood, and we have IV vitamins, or we have vitamin C in the blood. Now, out here in the blood, vitamin C is one of the strongest antioxidants. But when we give it in really, really high dosages, we've made the point that IV vitamin C comes into the cell and becomes a oxidant, okay, a pro-oxidant at high dosages. And we're going to talk about that in a second. But before we do, glucose, I just want to make the point that glucose is very, very similar to vitamin C in molecular structure. So there is a enzyme, there's a protein entrance way, and it is controlled by glucose 6-phosphate dehydrogenase. And this, so whenever you have glucose and IV vitamin C, they can compete for these entrances into the cell. So if you give a lot of high dose IV vitamin C, that in and of itself will compete with the glucose and will shut down the glucose metabolism temporarily for that cell because especially if the cancer is dependent on glucose, we know that IV vitamin C will compete with the glucose and it will inhibit, see right here, aerobic glycolysis. That's that glucose-dependent pathway that we talked about when glucose comes into the cell. So, and that's another reason why at the Rordan Clinic, they always check glucose 6-phosphate dehydrogenase before they start giving high-dose vitamin C, because if you have a deficiency of glucose 6-phosphate dehydrogenase and you give a whole bunch of vitamin C, it's not going to work. And so I think for those of you who are doing high-dose vitamin C, make sure that the doctor who is working with you has checked you for G6PD deficiency. Okay, so another main point is that Jane makes on, and I reference this, is page 392 in her book, that IV vitamin C alone is 10 times more lethal than the chemo to the cancer. And you may say, well, why? Because this thing will kill the stem cells. It will kill anything that the chemo has left over. Chemo will kill a bunch of cells like we showed in an earlier slide, but there's going to be cells that have lived. But this different mechanism of action, we are killing anything now. So that's something to take in consideration of. And if we use doxycycline before the chemotherapy, the doxycycline now makes the IV vitamin C 100 times more lethal than the chemo to the cancer. Again, referenced on page 392. You can look up the journal references there. How does this do that? Well, we know that doxycycline in a pre-treatment, before the chemo, or before the vitamin C, it blocks all three pathways. The glucose pathway, the fatty acid pathway, and the amino acid glutamate pathway. So doxycycline is a good strategy to try to make the IV vitamin C even more lethal. And this is potentially like somebody who just had chemo maybe the day before, and now they're going to get IV vitamin C, because there are going to be stem cells that have been left behind after the chemo. So again, we're not advocating to take the place of the chemo. If the oncologist feels like it's necessary, great, but we just want to try to mop up what's left, right? So what happens when this IV vitamin C comes into the cell? Like, what's the deal there? Well, here we go. We're in the cell now. The IV vitamin C is here. That's what this big fat C is supposed to mean. It's supposed to mean now the vitamin C inside the cell is much, much higher than usual. And when that occurs, the concentration of the C gets to a point where it begins to interact with the iron that is already in the cell. Now, the iron, believe it or not, don't ask me why, I'm still looking on papers for this, but cancer cells have much more iron in them than do normal cells. So when this high-dose vitamin C gets into the cell, it will now interact with the iron, and it will oxidize the iron. More like a pro-oxidant now. Remember, low-dose vitamin C out here in the periphery is an antioxidant. It's a good guy. Inside the cell, vitamin C becomes a pro-oxidant. It becomes a bad guy to the iron, to the cancer cells. Now, remember, there are good cells getting IV vitamin C inside them too. It's not doing anything in those cells because there's not any iron. There's not as much iron there. And so it doesn't do much there. But the cells that are taking this up that are cancerous with high iron, that iron is going to get oxidized. And that oxidation of the iron is going to generate hydrogen peroxide in the cell. Is that a good thing or a bad thing? Well, that's a bad thing if you're cancer, because now you're oxidizing, you're getting you're getting your butt kicked from the vitamin C oxidizing the iron because it's eating your iron up and it's creating hydrogen peroxide, and that peroxide is now acting like another pro-oxidant in the cell, and it's destroying the cell. And it's destroying these little lipid bilayer membranes that are all delicately put together to keep the cell in place. This cell's going to bust because it's got a bunch of hydrogen peroxide in there. Okay? So the vitamin C is blocking glucose metabolism. Okay? And it is increasing peroxide in the cells, and it is increasing cellular destruction, intracellular destruction of the cancer. Now, that's pretty cool. But another thing begins to happen that Jane points out, and we have to think about another thing. The cancer is not just going to take it. You know, if you hit it with vitamin C a couple of times, maybe let's say you have chemotherapy on Monday, and then on Tuesday you go in, like Tuesday and Thursday, maybe you go in for some IV vitamin C. Well, preferentially, what'll happen then is the cancer is going to try to survive, right? How does it do that? Well, another thing you have to remember is that in addition to the vitamin C here in the cell, the cancer has glutathione over here. Now, the healthy cells have glutathione too. The cancer cells have glutathione. Glutathione is the number one antioxidant that lives inside the cell. Vitamin C is the number one antioxidant that lives outside the cell. It's an antioxidant. It's a good guy. It's a donor. Just like a donation to the church. It donates. Glutathione is a donor. It's a good guy. It donates. But it's the main donor inside. Vitamin C is the main donor outside. But when the vitamin C gets pushed in high dosages, it becomes pro-oxidant and causes trouble for the cancer. But the cancer says, hey, you know what? I need to protect myself. So I'm going to use the glutathione that I have in the cell to save the day. So if the cancer cell has high amounts of intracellular glutathione, this glutathione can negate this oxidation of the iron and protect the iron from all this peroxide going on, getting made. So the cancer wants glutathione. So number one, when you are taking IV vitamin C, this is just another footnote, don't take a lot of glutathione. You know, like, oh, I'm going to help myself and I'm going to take antioxidants. Jane makes the point that you're not supposed to take antioxidants when you're trying to do high-dose vitamin C to kill the cancer, because the glutathione will negate the benefits of the oxidation of the iron. Just as an aside, the oxidation of iron is called ferroptosis, if you want to get fancy with the names, ferroptosis. Or we just want to stick with oxidation of the iron. That kind of says it all right there too. So, and just another little thing, when Linus Paulding started doing all his original research on using vitamin C, and I believe it was for cancer and for a lot of different treatments, a lot of the early researchers couldn't replicate what he was doing. And it started creating controversy about using IV vitamin C. And I think a lot of that controversy still persists to this day. But the interesting thing is that you've got to understand that using vitamin C for cancer or for anything else like this really depends on a bunch of stuff. Number one, you've got to have the right dose of IV vitamin C. Remember that kill zone that the Rordan Clinic was talking about? It's got to be the right dose to do the job. Well, you know, and a lot of the original research that people tried to replicate Linus Paulding, they were doing it with oral vitamin C. And oral vitamin C, especially when it's like in the form of water-soluble ascorbate vitamin C, it cannot get at high enough tissue concentrations. It cannot approach 350 milligrams per deciliter. It just can't. The normal concentration of vitamin C is like less than 1 milligram per deciliter. And you just can't get that high a dose with oral vitamin C. So that was a kind of a bummer. And then the other thing some of the early researchers were doing is they were giving their patients glutathione or other antioxidants as well. And so a lot of the early researchers couldn't replicate what he was doing. So again, just a little bit of history there to sort of make the point that glutathione is intracellular. It's there. If it is there, it is going to negate what you're trying to do with IV vitamin C. So what you want to do to try to lower intracellular glutathione concentrations is by using other things, and we won't get into them this talk, but sulfasalazine is one thing that will shut down this cysteine glutamate transport protein. It's called the XCT antiporter. You can just think of it as a garage door that lets the cysteine come in to the cell. The cell has to import cysteine to make glutathione. And if you use sulfasalazine, you're going to shut it down. And you're going to reduce intracellular glutathione. If you're using, there's another protein transporter. It's called the ASCT2 transporter, and it brings in glutamine also. And another agent, I believe it's cisplatin, will do the same thing. It will inhibit cysteine from coming into the cell. So that's a little bit beyond the talk, but just be aware that if anything, we do not want to supplement with glutathione. And if we are thinking about doing everything we can to lower intracellular glutathione, sulfasalazine is one of the main things. And there are some other things. Cisplatin will also do that as well. Another little ditty that I wanted to show you is this thing called the MDRP1, which is the multi-drug resistant protein 1. And that's that little thing here in blue. Now this thing is responsible, one of the things it does is it pumps out chemo drugs. So when your oncologists say if you have chemo on Monday, if the cancer is really good at proliferating or maybe having genetic mutations that allow this multi-drug resistant protein 1 to be replicated within the cell wall, like this picture is trying to show, it will pump out the chemo drugs. But research has been shown, and Jane has listed it, that mebendazole suppresses MDRP1 so that it shuts down the cancer cell's ability to pump out chemotherapeutic drugs, or many of them, which makes your chemotherapy even more deadly. Okay? Which is great. As a matter of fact, this is one of the mebendazole is one of the, it's been, use of mebendazole in cancer patients has really exploded to the point where, the price of mebendazole has pretty well skyrocketed because these drug companies know that they can charge these cancer patients an arm and a leg for mebendazole. And, it's really kind of sad in a way that these companies can do that to people who need this medicine so badly, something that has been shown to help. But that being said, Jane's book listed what she called a chemo cocktail. And me loving mnemonics like I love mnemonics, I just created the word, the phrase binds me. Like if you could think of chemo binds me. So if you're going to use chemotherapy, she says she advocated berberine, IV vitamin C, nonsteroidal, dipyridamole, a statin, and mebendazole, like a chemo cocktail. Now, that doesn't mean you negate everything else. You know, you can be on a whole bunch of different other repurposed meds, but these are the ones you want to emphasize. Why? Well, berberine shuts down oxidative phosphorylation. IV vitamin C, as we've just talked about how powerful that is after chemotherapy. And oxidative phosphorylation you think of as the Krebs cycle, the aerobic glycolysis, and all that. But when folks are fasting around chemotherapy times, the cancer may be even more fragile. Some of our patients have fasted a little bit before the chemo, during, and after the chemo. And if you can do that, there's research that shows how strong that is, or how strong fasting is in general. If you can only do fasting at certain times of the month, great. Some people they say don't do it during chemo, but there's been no adverse reactions reported from the literature that we've seen. And it's definitely an individual decision for sure, but the strategy is do everything you can do to weaken the cancer up to the point where you hit it with the chemo, and then after the chemo, you do everything you can to kill it after the chemo. And this is where the IV vitamin C comes in. A nonsteroidal plus a statin stimulates apoptosis. And remember, apoptosis is that programmed cellular death. In other words, the cell is dying when it's supposed to die. But cancer cells don't want to die. They want to live forever. So the apoptosis, and I have read places, and I'll have to create a slide set on this, but I believe that the apoptosis is sort of a genetic program within the mitochondria. Now remember, the mitochondria have their own unique DNA. They have their own genetic thing. And the mitochondria know when they're supposed to kick the bucket. And when the mitochondria die, that's sort of a theory on aging and a theory on cellular death. But nonsteroidals and statin combinations trigger apoptosis. So does dipyridamole and statins. I find that really fascinating. So Jane puts that in there because if you're having, like, as we said, chemo on Monday, and you're going to try to kill whatever's left over on maybe Tuesday, Wednesday, Thursday, or Tuesday, Friday, with IV vitamin C and maybe a little bit of fasting or whatever right in the middle of that, you want to stimulate the cellular death, apoptosis. Iodine also stimulates cellular apoptosis. And that's why I put this little iodine over here, because every cell in your body has a receptor for iodine. So there is something fundamental about iodine the cell needs. We were built, as our buddy David Brownstein says, our bodies were built to receive iodine. So talking about iodine beyond the scope of this presentation, but we want to use everything that we can during and after chemotherapy to make that chemotherapy more potent. I think I talked about everything on this slide a little bit, except this big fat T here in the middle. This is supposed to be tocotrienol. Tocotrienol is a form of vitamin D. It's the delta tocotrienol. It's found in the cell wall of the cells. So I just wanted to pay homage to delta tocotrienol. It is the most important glutathione is the most important intracellular antioxidant. Vitamin C is the most important antioxidant out of the cell, and tocotrienol is the most important antioxidant within the cell wall because why it is embedded in the cell wall. So I just wanted to say that, and we can talk more about tocotrienol at another talk as well, but I wanted to touch on all of these things. Let's see, I can't remember, honestly, if there's another slide. Now, let's see if there is. Nope, that's it. So we're going to stop here, and are there any questions? Haha, nobody can ask questions because I'm just recording this. So I hope this helps. Feel free to use this as sort of like maybe a talk to sort of like understand it a little bit more. We'll use these big pictures. Maybe I'm just thinking maybe we'll use it to drive home some important concepts so we're sort of all on the same page. So we can certainly talk about these transport proteins more and more. I didn't really intend to talk about them this talk. I just kind of threw them in, but we'll drill down on more and more of these as we go forth. So again, from Dr. Haley Brownstein and I, we wanted to say again how honored we are and humbled we are to be able to share this information and this journey with you. God bless you all, and we will see you soon.